Welcome to the Proteomics in Proximity podcast, where your co-hosts Dale Yuzuki, Cindy Lawley and Sarantis Clamidus from Olink Proteomics talk about the intersection of proteomics with genomics for drug target discovery, the application of proteomics to reveal disease biomarkers, and current trends in using proteomics to unlock biological mechanisms. Here we have your hosts, Dale, Cindy and Sarantis.
Welcome from the trade show floor of ASHG 2022.
That's the American Society of Human Genetics.
In Los Angeles.
Los Angeles.
And we have all three of us in the same place and at the same time.
So I have to remember not to look at you, but to look at you.
But we had and are having an awesome time.
Yeah,
and I want to start with talking about the workshop that Olink put on. So who wants to start talking about the workshop?
I'll do it.
I think Cindy should start.
Go for it.
Yeah. So the workshop was an industry session put on by Olink. And we thought, some people thought we would have only 50 people. I thought over 100. But we had over 200 people there for a presentation by Chris Whelan, who was discussing the work that Melissa Miller from Pfizer and Lyndon Mittenall from Regeneron spearheaded around bringing these groups together. That, several of these pharma proteomics partners came out of the exome sequencing consortium, but they brought on another 7 folks and then really built this proteomic data set that will be public soon.
What's Cindy's talking about is the UK Biobank Plasma Proteome Project.
Pharma proteomics.
I keep saying Plasma Proteome Project, but it's the Pharma Proteome Project.
It's all about the pharma, man.
And Chris, was spearheading it, he organizing it, and here it is, he's speaking at an industry workshop. So what did he talk about?
So he had several great points, but much of it was around this structure of building a pQTL data set for a systematic approach to therapeutic target discovery. But he also talked about a couple of examples within those data. And one I really liked was where he looked at stratifying with polygenic risk and then doing a biomarker study and using the genetics in a different way than integrating with looking at correlations to proteins, which I quite like those examples because there are lots of different ways that genetics can add value to how you structure your proteomics study to improve your power to detect the relationship between genetics and disease. So
here we had a full house, right? He's walking through the goals of the UKB Pharma Proteome Project.
Well done.
And then he had 54,000 samples worth of data. Did he talk really much about the data?
So he like I said, he dug in a little bit about examples, like PCSK9 was an example. I'm trying to remember if on this talk he talked about the ABO blood group. He often uses that. These are examples that are in that bioarchive preprint. And I'm gonna just give away a little behind the scenes thing I heard last night, and that was that the pharma proteomics partners were digging the idea of calling it the UKB OPP, be down with OPP, especially since we're in Los Angeles. So I thought that was fun, like putting Olink in there, but alas, not to be.
Olink Proteomics Project?
I did dig that whole link to the rap world. Sitting here in LA.
And when you're talking about last night, you were actually in a meeting then?
No, we actually had a celebratory dinner.
Oh, how neat.
Yeah, it was beautiful, really. To see these people I haven't met in person before and talk to them.
That's the advantage of an in-person conference, right? For many of us, it's like the first time in 3 years we've seen each other in person for ASHG, right?
That's right. And actually, since we're talking about the UKB OPP PPP, we actually have Evan Mills over here off to my left. I'm going to drag him in and have him just say a few words about. He went with me to that dinner. We had several people from Olink at that dinner. And so, Evan, can you just say hello and a little bit about your role in the
UKB close to the microphone and inside there we go.
Yep. I feel like I'm being led into the circle of trust here. So thank you so much, my friends. So this meeting's been incredible. My role, I'm the VP of our global strategic account team. And that essentially means, where is Olink going to make the most impact in the world? Trying to make sure we find those opportunities and work very closely with those partners. So the UK Biobank Pharmacoproteomics project clearly falls into that. And I've had the privilege of getting to know and work very closely with the 13 companies. And last night was a bit of a culmination where everyone sat around the dinner table, raised a glass, congratulations about the success of the project. All of the data is actually going to be coming imminently. So it was really one of those pinch yourself moments to be sitting with. I mean, there were so many thought leaders in the space, all around one table. You know, McCarthy, Slavé, Petrovski, Whelan,
Erin Smith,
Melissa Miller. I mean, it was just really an amazing opportunity. So yeah, it's been a heck of a meeting.
Yeah, it has. And while we're talking about Erin Smith, I will say Erin Smith's poster, she's at Takeda. So her poster here, which used proteomics to look at NASH, won a Reviewer's Choice Award. So I don't know if I'm close enough to the microphone to actually be recording this, but just saying that Erin Smith from Takeda, her poster that leveraged the UK Biobank data won a Reviewer's Choice Award, and so did Ben's son. Sorry, Erin Smith, Ben's son. So did Ben's son, and of course the Pharmacoproteomics partners are co-authors on that Sun et al. Paper.
Yeah. I mean, there's a remarkable number. I don't know if you've described the 19, abstracts and 6 talks. I mean, we are at a genetics conference and we're a proteomics company. So I just think this is a very important future viewing sign that the community that's used to ATs, Cs, and Gs are really opening up to large-scale proteomics and seeing the tremendous impact that it can have. I mean, Sarantis, as a multi-omics guy, were you surprised by this, or do you think this
actually, I'm surprised of how many people and they're talking about TK Biobank. I think the cohort is amazing. We have access and we have collaborations with a lot of academic pharma companies around these samples. And the future is here. The future of multi-omics is here. Now everybody talks about the next steps. Proteomics is now really nicely integrated with genomics. When they think about genomics, they consider immediately proteomics to integrate the data. And they're really great moments to come, And the question is always what comes next, right? And it was a discussion for the future. And it was really nice because I've been in a couple of talks where they tried to integrate, and there were discussions from Stephen Horvath, where they tried Steve Horvath actually, that they would try to integrate, thinking to integrate UK Biobank methylation profiles, methylation data, and really nicely explained that methylation data of plasma proteins, they really correlate with expression levels of the proteins themselves, right? And there are really nice correlation between protein levels and DNA methylation pattern more than the expression RNA levels. And that's something really nice and astonishing to hear. When you want to have a better view of your biomarkers, you have to integrate multi-omics approach, different layers of omics. And RNA itself doesn't say all the truth. Proteins are the bricks, are the blocks that correlate really nicely with changes that happen in DNA, genetics, DNA methylation, epigenetics. And that was a great take-home message from Steve Horvath, actually.
Now, Sarantis, you're Mr. Methylation here, right? And at a genetics conference, there's a lot of methylation talk, right? In terms of DNA, RNA, protein, and what's regulating the RNA. I mean, Stephen Horvath, right? He's talking in the context of UK Biobank, right? And he's giving this aspiration, wanting methylation data from the UK Biobank as another omic layer. And then what happens? All the questions are all around methylation. It's like, okay, we
People
know
are
about
excited. They're excited
we know
about that.
The Pharmaproteome project is going to have all these great drug targets. We know it's going to be great biomarkers and all this great stuff. It's like, okay, well, yeah, but we want something more.
Absolutely.
Yeah.
It's the multi-omics approach. That's the thing.
Yeah.
We are in the era of multi-omics, and I think proteomics brings a really big value to bridge different omics, right? That's the thing. Gives you precision and specificity.
I think that the tool building too. So I'll also say something else that was just a really exciting highlight for me was the Allen Award, of course, a very prestigious ASHG award that went to Professor Sir Peter Donnelly. He was knighted in 2019, I believe. But he has been pivotal since I started at Illumina during HapMap. So he was a a big leader and proponent for the approaches that were used in the HapMap project. And of course, that HapMap project had like one plate of Caucasian samples, one plate of Yoruba samples, and half a plate of each Han Chinese and Japanese samples, right? Just that was our first sort of swath at diversity across different ancestral populations. And after that, the 1,000 Genomes Project, once next generation sequencing became available. And so all of that documenting or cataloging of SNP diversity, he was pivotal in building out methods for analyzing those data. He built structure, right? So when I was in graduate school, Jonathan Pritchard was one of his students who built that program. I think he was a student. I don't think he was a postdoc. I think he was a student.
And this program Structure does what?
Structure actually is, I still see it in most sessions, in at least one talk in most sessions, the use of this amazing tool to essentially take data and use the data for it to generate the number of groups that are represented by the genetic data, right?
So these are these dot plots with the different colors. That's right. Where you have like African in this dimension, that's right, East Asian in this dimension.
Exactly. Yeah, beautiful, colorful plots. Yeah, that demonstrate sort of within this, we think there are 3 groups, or within the data, right? Letting the data speak for itself rather than a priori saying, like, and for me, looking at fish, I was, sampling from 8 locations, but if those 8 locations actually only represent 4 unique genetic entities, that says something about how to conserve or manage those fish. But let's go back to Peter Donnelly. I just want to say what a rock star he is. And then we can, I just
go right ahead.
Really want to call out to this man because he is, certainly a statistical geneticist, a statistical analyst, right? But with having that skill, but also being charismatic and able to bring people together to help them see what's possible with it, like being able to communicate. It just comes down to communication so often. And he's such a thought leader here. And so I was just so excited to see and when he told me he was getting an award, and then I looked up what he was getting an award of, I was just like, he's so humble and so amazing. So, now
the importance of communicating, right? The importance of working with others. The point I was going to make before was as far as the use of this term structure, this particular program structure, is that in the UK Biobank, people can say, I identify as I believe my parentage is Japanese. I have every reason to believe it. But genetically, could it be that there may be some Mongolian in Dale's background? Who knows? Right?
Yeah.
But then a program like this, right? Say I sign up for All of Us, right? And by the way, right, I went to an All of Us session. And it was fascinating to see that, yes, even though COVID kind of slowed their enrollment down and slowed the progress down, they're up and going again. And so I went to joinallofus.org this morning and I started the enrollment. Why? I think they're only about 500,000 into their 1 million sample cohort. And like you mentioned, Cindy, right? They're working on getting great diversity. There was one talk that talked about engaging Native American, American Native populations, and they're really emphasizing that. And there was one particular talk where it was just how many tribes in just Alaska? It's like 211. It was wild, right?
Well, there's, what is it? How many ethno-linguistic groups in Africa, right? 54 countries, but like 2,000, maybe it's 3,000.
And you remind me, right, of another great plenary session, right, where they're announcing, right, genome centers of excellence in Africa. They're getting a conference in Cape Town in the spring. Francis Collins kicking that off.
We have an abstract submitted to that conference, by the way.
Oh, how cool.
Yeah.
So
we hope.
The emphasis, right, on non-European populations, the emphasis on diversity, the emphasis on African genomes. In that one session, they talked about Lassa infectivity. They talked about sickle cell anemia and just
HIV co-infections
and some remarkable disparities, right, in terms of the number of, for example, for sickle cell, right, there are several gene therapy trials here in the U.S. Any in Africa.
Yeah.
0.
I'd like to double click back on the All of Us program, because something that I learned here this meeting was that it isn't currently set up in the way that UK Biobank is set up with an international sharing structure, which, I imagine they have good reasons for, but I think there's a hope that All of Us will consider creating an international sharing structure that will enable if we build these centers of excellence in Africa, we want those scientists to be able to analyze those data or interplay with those data. Maybe it can't be transferred or downloaded, but having a federated sharing environment, having a protected sharing environment that allows insights from those data to then be transferred, because those investments can be used globally, like the UK Biobank project has.
I mean, there was a tweet, right, that was really popular talking about how frequent the UK Biobank is popping up. Somebody saying, they felt like every other talk mentions the UK Biobank.
It's a great validation cohort, right? But also for initial findings. People are still mining those data directly. I mean, as a grad student, I would imagine that's a rich data set to have access to.
And a lot of sequencing, right? And I know that you're looking really close to the new sequencing technologies. Would you like to share some information about that, actually?
Yeah, well, I mean, Maroon 5, right, was playing the
we
first night.
We should talk about Maroon 5.
Yeah, I mean, okay, so it's a gigantic line. I mean, Olink had our own VIP event. It was pretty small, right? But then all these people lining up to hear Maroon 5 and pictures and stuff like that. But why? Why have, a popular band play? It's because they announced 2 new platforms, PacBio, 15x, the Revio 15x, the throughput of a Sequel 2, the Onso, which is the Omniome short read platform. You've got MGI here, you've got Ultimate Genomics here, we've got Singular Genomics here, we've got Element Biosciences here, all with new instruments, right? All sort of bringing it to market.
With their own differentiators,
Their own differentiators.
And, what about the cost? What do you think with the cost?
Just driving it down. Now, for what does that mean for Olink? It means great things for Olink. Why? Because Olink customers receive the benefit of those lower costs.
Exactly.
That they're not held to $6 a gigabase for NovaSeq 6000. NovaSeq 6000 SP4, right?
I have to insert the company line here and just say we are not validated currently on any of these platforms, but we certainly have collaborations with many of them.
Yes, and we had a press release.
With an interest to keep a focus on, how do we continue to drive down cost in genomics? We see the benefits. Yeah, that's so important.
Yep. And, go ahead.
I'm sorry, go ahead first.
Oh, well, the NovaSeq X, right? They're announcing $2 a gigabase at the end, toward the end of 2023, which is fine, and a new instrument which they have right here in the Illumina booth. But you walk over to MGI, right? The BGI, MGI, they've got a T7, $1.50 per gigabase now. And you just say, well, wow, that's pretty remarkable. That is happening here at this conference, right? Because of all the different progress with lawsuits and what have you, freedom to operate. But nonetheless, right? Ultima saying we're going to be at $2 a gigabase, come February of next year. I went to the Element talk and it was remarkable, right? How they're driving down costs and how they have really high quality. So one of the interesting things here at ASHG is science communication. And of course, with posters, there's some 3,000 posters, and it's a remarkable work being done in lots of places. We have this thing in our booth, right, with a number of different posters from the UK Biobank, but I was looking for Cindy's poster and I couldn't find it because there's so many people crowded around it. So Cindy, why don't you tell us a little bit about your poster?
Yeah, it was a bit of a surprise to me, actually, because there are so many posters. I've not had a crowd around an ASHG poster like this
How interesting.
In any of the times that I've presented here. So
that is so cool.
It was really cool. It was cool. So thanks for noticing. And someone took a picture. I thought, wouldn't it be great if someone took a picture and then there's Dale taking a picture. And I was thinking, I should have worn my other shoes. But anyway, the poster is really an aspirational poster about the work being done by the UK Biobank with proteins, meaning that I talk about a previous proteogenomics project. It's a little smaller scale on the protein side, so 30,000 samples, so respectable sample size. It's a cohort of cohorts you're bringing together. This is the scallop study, and then 90 proteins, right? And so I talk about the 90 proteins, how many pQTL discoveries were made possible by the 30,000 samples, and then how many of those from Mendelian randomization were able to have strong evidence for potential therapeutic targets. And so if we saw that same sort of 5.5% conversion to potential therapeutic targets in the UK Biobank, apply that to just the 1,500 proteins that are published in the bioRxiv paper, that's over 500 potential therapeutic targets, disease agnostic across different diseases. So anything that's going to give pharma a more unbiased approach or a disease agnostic approach to and evidence to support consideration of particular proteins as targets, I get excited about that. So there were several people that came, and I think maybe because of where it was located, others that were walking by stopped as well. And so I got to present it several times, which is really fun. But an hour and a half is not enough time for for a poster session where you want to see even just 5 or 10 posters of 3,000 posters, right? Because there's just so much space to cover, and you want to have a conversation with each person presenting it. And so it was just nice that people took the time to come and listen.
One particular poster that caught my eye was one from Takeda, right? That uses UK Biobank.
Yep. That's Erin's. Yeah, I think we mentioned that when Evan popped his head in. But that one that got the Reviewer's Choice Award. Such a talking about the potential for these proteins to suggest something about the action in hepatocytes. What are exosomes they mentioned in the conclusions. Really for future studies, which is such a hot topic that people are talking about, but I'm not seeing a lot of publications yet coming out looking at broad-scale proteomics within the exosome. That's.
Because I think it's quite difficult also, not only to isolate, but also to extract proteins from that. But we are working, and I think we have a lot of now case studies that we are moving in this direction, right? From all the proteomics, it feels really nice to actually some studies, actually.
That's where the small input volume, right, Sarantis?
Super low amounts of protein. Olink being able to pick up protein, right? Yeah. Protein signals from a remarkable number of sample types. And, here, we're here on the show floor, here there's so much going on. You can hear overhead. They're saying that the show floor is opening up in 15 minutes.
Are tucked away, and your booth looks neat and professional for the last show day. Thank you.
Last show day.
The last show day. Well, any final thoughts? Here from ASHG?
What I can say from my side is we see a lot of need for liquid biopsies, for non-invasive methods, and we see that they are correlating really nicely, especially in the case of cancer with solid cancer. People in the future, I hope they will not need to go through solid cancers.
Mm-hmm.
Yeah, I'd say mine is, the promise of successes of pre-competitive collaborations, bringing together groups to have power for large studies. I think every time we see a success in this regard, it lays the groundwork for future potential successes. And I think if we can get the lawyers to all agree on terms, then I think that's the hard part, is to make sure that the interests of each of the partners is respected and they're able to get benefit from it. But I'm very optimistic about that.
It is optimistic time. Yeah. Well, thank you for joining us.
Thank you very much.
Thank you for listening to the Proteomics in Proximity podcast, brought to you by Olink Proteomics. To contact the hosts or for further information, simply email info@olink.com.